welcome back to the next lesson. This is going to be section 16.5. The section title is Focus on the Human Body, and it's going to be about neuropeptides and peptide hormones. I noticed the chapter has quite a few of these focus on the human bodies, well, usually about 2 or 3 per chapter, and they're kind of a neat little break to put the material into context, because this is biochemistry, and so we're studying molecules that chemical molecules in the body and stuff, but we need times to really put it into perspective and give a more zoomed in perspective of how this stuff applies to the human body. And so whether it has to do with real medicines that have been developed or things like that, the book gives these little focus on the human bodies, which are kind of nice. And so we've discussed peptides. That was the last lesson, 16.4. And so now we're going to focus on some very specific peptides. Okay, so you might be wondering, when are we going to start talking about proteins? This whole chapter is about proteins. This unit is on proteins, and proteins are polypeptides. So we've done a lot of talk so far because we really need to understand what polypeptides are and what they mean. And so we had to first start our conversation about amino acids, because amino acids first make up peptides. We've studied that there's dipeptides and tripeptides so far, but we're going to make long chains of peptides eventually, and we'll talk about protein folding, and we'll get there. But since we just talked about, again, peptides like dipeptides and tripeptides, those actually in and of themselves, before they're connected to make long proteins and such, they can have use in our body as well. So let's look right away into, sorry for the tunnel of doom there, let's look right away at a couple. 16.5a discusses neuropeptides, enkephalins, and pain relief. So enkephalins, they're peptides that are synthesized in the brain. So that's where they're going to be manufactured, is in your brain. And they act as painkillers and sedatives by binding to pain receptors. So oftentimes we take medicines to do that, but your body has its own way of achieving the same results by molecules that will attach to these pain receptors. Okay, because those pain receptors are in your brain. The two enkephalins that differ are two of them that differ in identity of only one amino acid are known, that we know of. That is interesting. They only differ by one amino acid. There is met enkephalin and leu enkephalin. All right, and it all has to do with the C-terminal. It all has to do with the C-terminal amino acid. You remember how we talked about for peptides, you have the N-terminal, where the nitrogen is on the terminal end, and then you have the C-terminal, where the carboxylic acid, the carbon, is on the terminal end. Well, the amino acid that has the C end, the C-terminal, if it's methionine, then you'll have met enkephalin, M-E-T enkephalin. If on the C end you have leucine, if you have the amino acid leucine here in red, you see there, then you have leu enkephalin. All right, and so here you go. The rest of the molecule is the same. You have this amino acid, which is methionine. Tyrosine. Excuse me, I'm trying to look at that like that's not methionine. This is tyrosine. You have tyrosine. Okay, and you can see it in the name here. They have the abbreviations there. So you have tyrosine, glycine again, PHE. Make sure I say that right. Go back to my list. Let's look at this list. Phenylalanine, yeah. Phenylalanine. Not to be confused with alanine. Phenylalanine. Remember, phenyl is this ring structure. That's what that means. And then finally, you have methionine for this enkephalin, but you have leucine for this enkephalin. So you either have met enkephalin or leu enkephalin. Okay, now let's talk about what the main difference is. Well, right away you can see a color difference. I like how they show the ball and stick model. This is kind of cool. It shows you all the bonds, all the atoms, and really shows you its three-dimensional structure there, like kind of what it looks like, its shape. And so right away again you can see a color difference. I can see a yellow atom here, which I don't see here. So basically there's a sulfur atom in this one, and there's no sulfur in this one. So that's one of the key differences, but there are other ones that we'll look at here. The addictive narcotic analgesics. Analgesics is a word for pain medicines. And the addictive narcotic analgesics, morphine and heroin, they bind to the same receptors as the enkephalins and thus produce a similar physiological response. Enkephalins are related to a group of larger polypeptides called endorphins that contain 16 to 31 amino acids. Endorphins also block pain and are thought to produce a feeling of well-being experienced by an athlete after excessive or strenuous exercise. You know that good feeling you feel sometimes after you get your heart rate up and get your muscles moving? That's called those are caused by endorphins released into your bloodstream. Okay, all this is manufactured in your brain and released into your bloodstream, and so then once they're in your blood, they attach to receptors that are waiting for these. The receptors are like the lock, waiting for the keys, and the keys are the endorphins, and they will trigger that response. All right, and so problem 16.12 says label the 4 amide bonds in metenkephalin. So remember what the amide bonds are. They're always when you have carbon bonded to a nitrogen, but it's real important that you remember which carbon and which nitrogen you should be referring to. And it's always if you're looking at my pointer, it's this one right here. I first kind of went to this one, but no, that's not it. It's going to be the carbon and nitrogen, because you see this nitrogen is bonded to 2 different carbons. So the key is remembering that it's where the nitrogen of one amino acid bonds to the carboxylic acid of the other. And so that would be right here, right? This is the carboxylic acid part of this amino acid. And this would be the amine part of this amino acid. Okay, so you're always looking for that amide bond. And if you have trouble with that, go back to here. Remember, amide bonds are just general terms. The peptide bond is a better term. It really is a better term. And again, it's going to be this carbon-nitrogen bond, but close to this carboxylic acid part. All right? So it's close to the carboxylic acid part because it's the carboxylic acid of one amino acid coming together with the amine part of another amino acid. And after the water is released, because remember you take off an oxygen from the carboxylic acid and you take off 2 hydrogens from the amine, you get this peptide bond, and then you lose a water. All right. Another one that is interesting is or another type of peptide that's in the body are peptide hormones. And these are 2 that get a lot of attention. Is oxytocin and vasopressin. Oxytocin and vasopressin. And they're cyclic peptide hormones. Do you see how you have like a cyclic structure here? Right? You kind of have a full circle situation going on here. I mean, you also have some branching there, but you have kind of a cyclic structure. And so there's cyclic peptide hormones secreted by the pituitary gland, and their sequences are identical. Oxytocin and vasopressin, they have identical sequences except for 2 amino acids, and it's the N-terminal amino acid. So check this out. I had to stare at this for a little bit because I ignored this, the disulfide bond part, but I was staring at this thinking, wait a minute, how can this be the N-terminal when it's not really terminal? You see how cysteine is like part of this ring? It's part of this ring. So this is obviously a terminal one, right? This is obviously a terminal, and it's another thing I want to address in just a second. But where's the other terminal? Where's the other terminal? Well, it is this cysteine. This is the end of the chain. You see right here, you don't have an amino acid. This is not an amino acid. This is not a peptide bond. This is a disulfide bond, which we're going to talk about this in the next few lessons. This is an amino acid bonding to another amino acid, but not through a peptide bond. Not through a peptide bond. So as far as what's exposed here, there is still actually the amine group exposed. And so that is your N-terminal amino acid still. Now the C-terminal is actually still glycine, but interestingly enough, there's also an NH2 there. But don't confuse that with thinking it's the N-terminal just because there's a nitrogen there. Glycine's amine group is connected to the leucine carboxylic acid group. Like here's the peptide bond. So glycine does not have its amino group or there's no N group that's terminal. It's the C group that's terminal. So you have a C-terminal amino acid glycine here, but there is an NH2 that's attached there. So we'll talk about that in just a second again. But oxytocin is the first one we'll look at. It stimulates the contraction of uterine muscles. So for the uterus is where embryos develop inside of females. And you obviously got to get that embryo out, which I was a witness of recently. The first time I've ever witnessed that. And in order to do that, you need those muscles to contract. And so if you ever hear of females saying they're getting contractions, that's what they're talking about. And I had to actually witness my wife go through a lot of pain for the few hours or a lot of hours before labor until she got her epidural, which acted as an analgesic. They blocked pain receptors because it was a little intense. But for the first few hours before we got to the hospital, she was having some painful contractions. She could hardly even stand up, couldn't walk. And they come every, minute or so, or every couple minutes. About 5 minutes in the beginning. But so oxytocin is what's responsible for that. And you can actually inject it. If you wanted to stimulate labor, you could do that. You could stimulate labor by injecting oxytocin under the trade names Pitocin and Syntocinon. So they're used to induce labor. If a female is having contractions and the baby's not coming, you got to induce that labor. It could be a risky situation later on. It could be a risky situation. So sometimes you need to induce those labors, or if the baby's just been in there too long, after so many weeks, they finally say, we got to induce this, because just like you don't want a premature baby, you don't want a baby that, you want babies. I'm saying just like you don't necessarily want the baby to come out premature, you don't want it to come out post-mature either. I don't know if that's the right word for that, but vasopressin, all right, is the other one. This is another type of peptide hormone, kind of like oxytocin, in the fact that they both have the same structure, except for right here, this one has phenylalanine and this one has leucine in that corner there. All right. Vasopressin is an antidiuretic. Well, first you got to know what diuretic means before you know what antidiuretic. So an diuretic is something that makes you lose water. So it makes your body, it makes your kidneys secrete urine, or it makes your kidneys secrete water, and so you end up with excess urine in your bladder, and so you got to go to the bathroom a lot because your kidneys are getting rid of a lot of fluid. And so vasopressin is an antidiuretic, or ADH, which does the opposite. It targets the kidneys and helps to keep the electrolytes and keep body fluids in the normal range. And so what happens is vasopressin is secreted when the body is dehydrated, thus causing the kidneys to retain fluid. All right, so you won't go to the bathroom as much. And so in essence, it decreases the volume of urine. Okay, that's the opposite of what a diuretic does. A diuretic increases the volume of urine because your kidneys are constantly releasing liquid. All right, and so the N-terminal amino acid in both hormones, again, is cysteine residue. It's a cysteine residue here, the N-terminal. And the C-terminal residue is glycine. You can see that here, GLY, that's the C-terminal. Now, this is an important point I wanted to bring up again. Instead of a free carboxylate, right, because this is the C-terminal, but instead of a free carboxylate, both peptides have an amide at the C. Both peptides have an amide at the C-terminal end. So this is indicated with an additional NH2 group drawn at the end of the chain. The structure of both peptides includes a disulfide bond. It's a vocab word, the disulfide bond. And it's a form of covalent bonding. This is not just like a hydrogen bonding or an electrostatic attraction. This is actual chemical bond, sharing of electrons. It's a covalent bond in which the thiol groups, or SH groups, sulfur hydrogen groups, form two cysteine residues in which they form two cysteine residues are oxidized to form a sulfur-sulfur bond. So you see how you have a cysteine and a cysteine? The two cysteines are bonded to each other by that sulfide group there, by that SH group, right? And so in oxytocin and vasopressin, the disulfide bonds make the peptides cyclic. So the reason these are cyclic is because of those disulfide bonds between these cysteines here. Cysteine bonds to cysteine, literally, via that disulfide bond, causing a ring structure here. All right, and so here's the next little picture again. Oxytocin induces labor by stimulating the uterus to contract. So it's made in the brain, but it stimulates the muscles of the uterus. Okay, so it travels through your blood. After delivery, oxytocin stimulates weak uterine contractions so that the uterus may return to its normal size. So the uterus is still going through those contractions every so often in a periodic way. Maybe not every 5 minutes, and obviously not as strong. These are weak uterine contractions so that it goes back to regular size. It also causes muscle contractions in the breast, making breast milk available to a nursing infant. And so that also may need to be done artificially by an injection to help stimulate breast milk, but I think there's other ways. They don't really do that. But that is a challenge, and something I've experienced too recently. Not myself personally, but I've witnessed that, feeding Levi the first week or two was kind of a challenge because my wife is pretty not stubborn. I don't want to use that. Stubborn, but I want to use that in a good way. She's trying to steer away from formula and really stick to the breast milk because of all the hormones and antibodies and things that are in there. And so it's just way better than formula. And so she tried and kept trying. And it's just, you don't get as much milk in the beginning, but I think things are going pretty good now. So we're good. But that hormone is responsible for making breast milk available because the muscle contractions need to happen to get the milk to move. All right. Secretion of vasopressin by the pituitary gland, which is also in the brain. It's a small gland inside of the brain here in the midbrain. It causes the kidneys to retain fluid. Remember we said that? It's the opposite of losing fluid, retaining fluid, resulting in decreased urine output. All right, so the 16.6 is now about proteins. I'm not going to start that lesson yet, but just wanted to show you the chemical structure here again for oxytocin. A lot of this, it's the love chemical, right? Oftentimes it gets that name, and I have a picture of it in my lab. I know you guys have seen it back there by the sink. You've seen the structure of oxytocin, C43H66. Quite a large molecule there. And I also looked up oxytocin or pitocin on Wikipedia. There's some pretty cool stuff to find. Here's vasopressin on Wikipedia also. And here's something interesting I wanted to show you about vasopressin. Check this out. A third function is possible. So these two functions about vasopressin, about the kidneys, they discuss that. That's kind of the primary. But a third function is possible. Check this out. Some AVP, which is arginine, vasopressin, may be released directly into the brain from the hypothalamus and may play an important role in social behavior, sexual motivation, and pair bonding. And so what is pair bonding? So I clicked on that, and this is an interesting term that's frequently used in sociobiology or evolutionary biology circles. And it was some research done in the 40s. So this term pair bonding was coined in the 40s when some research was done about animals and their social behavior. So pair bonding is when you get like a monogamous relationship. So I don't know if you've heard of voles. They're small rodents and like hamsters or whatever. I'm not sure if that's the genus. I think it's the genus. It could be a family, whatever, but it's a group of organisms in the taxonomic rank that have shorter hairy tail, slightly rounder head, smaller ears and eyes, and basically you can have monogamous voles. Monogamous means only one partner, and they have a greater density and distribution of vasopressin receptors in their brain when compared to polygamous voles. So these differences are located in the ventral forebrain and the dopamine-mediated reward pathway. So the idea is that these organisms that perform monogamous relationships have extra vasopressin receptors, suggesting that it would play a key role in that. All right, there you have it, 16.5. Told you that was going to be a short video. Bye. Stay tuned for the next ones.